Hi, everyone. I see that people are still logging in. We're about a minute away from starting, so we're just going to wait for a few more people to join us, and we'll get started in a few minutes. Okay, it's 3 o'clock. So I am going to move to start this. Let me unmute my panelists. Okay, so making sure all my panelists are unmuted. So we're going to start now. So welcome to the LABEST Presents COVID-19 Spotlight Series. My name is Marvi Valcourt, and I'm in charge of the marketing for UCLA Technology Development Group. We are the division that manages the intellectual property for the university. Before we get started, just a little housekeeping. This webinar is scheduled to last about an hour. We're going to break that up by having about 45 minutes of discussion and then hold back about 10 to 15 minutes for questions. If the attendees can please use your Q&A function on the Zoom bar to ask your questions, and my colleague Daniel Morton will be taking a look at those and preparing those for when we get to the Q&A section. So now I'd like to turn it over to Mark Wisniewski, my colleague at TDG.
Thank you, Marvi, and welcome everyone to our webinar. I'm Mark Wisniewski, Senior Director of Biopharmaceuticals at UCLA TDG. And in the spirit of our annual LABEST conference, this is our inaugural LABEST Presents webinar. And the goal in the spirit of LABEST is to promote awareness of leading edge translational research in order to facilitate relationships and ultimately collaborations between the panelists and also our bioscience stakeholders in LA and globally. Today's discussion is to focus on two critical areas for addressing the COVID-19 pandemic, which are detection and tracking of COVID-19. So thank you again, Marvi, for moderating, and our panelists for participating, and the TDG team who helped organize this webinar.
Thank you, Mark. So we're going to start today with having each researcher introduce themselves and do a summary of their work as it relates to COVID-19. Unfortunately, Dr. Otto Yang is not available to join us today because he's busy with clinical trials. So with that, we're going to start with Ren Sun.
All right. I'm a professor in Department of Pharmacology and the Bioengineering, and I have been at UCLA for 23 years working on virus. And if you can show the first slide, so I can get into the research work we're doing on COVID-19. The diagram of the curve of viral replication indicated by the red, and then the antibody response in green and blue. And in the first several days after virus infection, the virus replication increased dramatically before the antibody are produced. And that's a period of time the infected person may not be symptomatic, but it has a very high level of virus and are the major media of transmitting the virus to other people. And it's important to detect the virus before the antibody response occurred. And there are two ways to detect the virus traditionally. One is using nucleic acid measurement, and which is current standard measurement, but which requires extensive physical and manpower. And then there are easier assays, which is detect antigen, use immunological assays, which usually requires antibodies. And currently, there are not many useful assays for the antigen yet. And what we are trying to do is use simpler approach to produce antibody mimic, and it's called monobody, and which is diagrammed on the right side. And there is a protein structure that's the smaller protein we're using as a scaffold to generate antibody mimic. And there are two loops, a red and a blue. It's labeled BC and FG. Those two loops will randomize it and make it very, very diverse. And some of the conformation will be able to recognize the protein from COVID, from SARS-CoV-2. And the protein we target is N protein, which is the most abundant and stable protein in the serum and in the saliva. So we can potentially recognize the protein in the infected people, and using simple immunological assays, such as ELISA. And at the bottom of the slides, and there are several slides that indicates the infected cell, and the red panel is using our newly generated small protein, and we temporarily named NN3, and we can use that to recognize the viral protein in the COVID-2 infected cells. And the other diagram is the detection of the N protein using our monobodies on ELISA assays. And we can detect the protein to several picogram per mil sensitivity. And this is where the current research stands in the laboratory. And we try to stabilize the protein at different conditions, and so make it robust in different platforms of detections. And currently, one issue we are facing is the protein is not robust in different conditions yet. Okay. So we're looking for collaborations to make the protein stable. Meanwhile, we are make modification of the protein, and another approach is collaborate with other investigators, and you will see Dan will mention that. Another approach is using different condition of detections. All right, I will stop here.
Thank you, Ren. Daniel, would you like to talk about your work?
Sure, yeah, if you could go forward on the slide. So I'm Dan Kamei. I'm a professor in bioengineering. I've been here almost 17 years, and the last decade or so, we've been working on developing point-of-care diagnostics. And our approach has been to use engineering science, so try to develop models whenever possible to complement our experiments so that we can make these robust technologies that are predictive. So just talking about two of our very, very recent successes. One of them is with Dr. May St. John at the UCLA Medical Hospital, where we developed a point-of-care kit to detect leaks of cerebrospinal fluid. And so you can see the kit there with instructions, and we validated this with patient samples. So this is one of the successes. Another recent one is related to a startup company that I co-founded. At this company, there are 4 of my alumni there, lab alumni. It's called Phase Scientific. And exciting part is a couple weeks ago, they did a commercial launch of Phasify, which is an RNA extraction kit for COVID-19. And so this is really important because, you know, one of the limiting factors of getting a lot of people tested is, you know, this RNA extraction kit is in shortage. And so if we could help and alleviate that, we can help test more people. And then connecting to Dr. Rensan's work that was just mentioned, it's really exciting. You know, he sent me a preprint of the paper, and I'm really amazed at what his group has done in only 2.5 months. And so we're now working with him and using those monobodies to put them on paper and try to make a lateral flow immunoassay.
Thank you. Now, Eran, would you like to move on to your work?
Sure, yeah. Can you show the next slide? Yeah, okay. So I'm Eran Halperin. I'm a faculty in computer science and the School of Medicine. So in a couple of departments over there, including computational medicine. So basically my background is actually in more on the computer science side, but I've been working the last, I would say 20 years on computational biology and computational medicine. And specifically, some of the projects that we're looking at are using the data from the hospital, the electronic health records, or other data that is that are kind of public or from collaborators that are related to the hospitals, in order to improve patient care. Once we basically saw that the COVID-19 is kind of starting to take place, we thought of basically trying to kind of move some of the project that we have into that space. And we have a few projects that are going on this. All the goals in all of them is essentially forecasting in different levels. So one of the levels is essentially just the level of the patient. The patient comes in, you want to know, the COVID patient comes in and you want to know what's going to happen to the patient in the next few hours, in the next few days. And that's essentially based on the electronic health records. The other one is basically trying to understand what is going on in terms of the spread of the of COVID in specific areas. How is it going to affect the hospital, the system, and basically you want to know how this is going to be affecting us in the next couple of weeks, and obviously, ideally in the next few months. But definitely, when you think about what you need to decide now about what to do locally in the hospital, locally in the region, or locally in the country, is based on what you think that is going to happen in the next few weeks. One of the projects that we launched is called Stop COVID-19 Together. And that's the website that you see here. And basically what it is essentially a survey that we ask people to fill out and give us information about essentially the symptoms, exposure, and things like social distancing and other measures that basically people are taking in order to be able to figure out if this can be used. So first of all, in order to be able to know what is going on right now, but also in order to be able to show what will happen in the future. Can you go to the next slide? I want to show a couple of slides just for examples of what you could potentially get from this. So what you see on the top left is a picture that from just kind of a screenshot from the web page. And so in this case, you're looking at the level of cities, and you basically go to the city itself and you see what is the, first of all, you see information that comes from the publicly reported. So in this case, let's say in Santa Monica, we have 107 cases, and there's no deaths that are available, at least the information is not available. And then we also give you information about what's going on in the survey itself. So from the people that we see in the survey, we have about 30% of the people that had at least one symptom that is related to COVID, and 3% in this case that were tested for COVID. And then most people in this area are physically distancing, which means that basically they said that they would like to do the physical distancing. They're basically gonna adhere to this right now. And so this is just an example when you click on one point, and we add more and more data on top of this based on the information that we get. And the resolution that we can get to depends on the number of people in that area that answer these questions. What you see in the right, bottom right picture is what is going to happen based on the predictions that we have in terms of the resources of the hospital. So in the examples that you see here, you can try to predict what is the number of ventilators that we're going to use, what is the number of people that the ICU beds that we're going to use, and what is the number of people that are going to be in the hospital in the next couple of weeks. And again, this is very critical if you want to plan what to do with the hospital in terms of, let's say, if you decide that you want to put people, take cases that are more urgent or less urgent, depending on how many beds you actually have and how many resources. Another example, you can go to the next slide. Another example is something that we just added recently. We asked people on questions about the places that they typically go to. So specifically these days, because of the stay-at-home order, so basically what we have is that most places are closed, and people typically only go to the grocery store or maybe to the pharmacy. So we ask people which grocery store did they go to, and then for each grocery store we can see what's the number of people that that are reported to be sick in that hospital, and we give it as a frequency, and we only report when we have enough numbers. So not all the grocery stores are going to be there, but the LA area is pretty well covered. So you can basically see the red points here are essentially the ones that have more than 10% of the people that reported they have some type of sickness. And then if you click on each of them, you'll see that some of them actually have 30, 40, 50% of the people that go to the grocery store are sick, and some of them are just 4 or 5%. So that basically, so again, this kind of information, even if you just look at it just visually, you can see that there are some clusters, and we're basically taking in the back end the data, and we're trying to figure out where these clusters are going to. So just the dynamics of these clusters. And we're asking, this is actually something that we're starting today, we're basically asking for follow, so people can add their email address, and then we can ask them for a follow-up question. So we ask them essentially, again, for 2 or 3 questions. One of them is going to be, have your symptoms changed? So basically, what are your symptoms today? Because maybe a week ago when you filled it out, you felt okay, but now you don't, or vice versa. And that will allow us to see the kind of the dynamics in terms of time and space of the spread. So that's it.
Great, thank you so much. So now what we'd like to do, I'm going to unshare the screen here. What we'd like to do is talk about collaborations. And so I want to move to Eran and Daniel and have you talk about your current collaborations, and then afterwards we can talk about potential collaborations between the group.
So maybe I'll start, and then Ren could add. So we received proteins pretty much just this Saturday and started doing some runs already. So this is off and running, and every time we have a question, Ren or his student responds very quickly. So this has been, I don't know, I have high hopes for this collaboration.
Yeah, I want to add the technology platform. Dan is a really good at it and have pioneered many improvements with that platform. I'm very hopeful, and if this technology can be used on Dan's platform, and it will be a huge step forward to control the spreading of the virus, because the platform Dan using enable detection in 10-15 minutes, and it can be done in the train station and in airport or at a Whole Food market or at home, and at any point of care. So anybody can use it. It's similar to the pregnancy test, and you look at the test strip, and whether 2 or 3 line of mark will show up. So that is really going to be a powerful tool to control the virus spreading. And maybe I can add one more with Eran, and if we can have this point of care detection, and if those items can be added to the survey, and then we add more specific information to the survey, and then those data will be very precise and useful for reporting and for data processing to show where the hotspots are.
Yeah, maybe I should add to this. So one of the motivations for the survey is that the data that is out there is really noisy and problematic. And one of the biggest challenges is because there's no, I mean, I guess everybody knows it by now, but there's no good detection, or there's not enough kits. And so there's probably a lot of people that have COVID that we don't know of. And even though they might not be very severe cases, so they don't show up to the hospital, they do spread the disease, which is less of a problem today because we're all staying at home. But as soon as we go back to, to the schools, and the kids go back to the schools, and we go back to our offices, then these people are still going to give to be contagious. And so this is, essentially these kind of projects that basically trying to track the information are important now, but they're going to be even more important if this is basically going to stay around for a long time, because then we'll just need to understand what is the situation at any given point. One of the ways to kind of combine these two things is, like Ren was saying, if you have a detection mechanism that is, so basically if you have a kit that is very easy to use, and scalable, people can basically test themselves periodically, or go to the clinic and basically getting tested, and then they basically can add it to the survey. If you look at the survey results right now that we have, most people haven't been tested for COVID. So when we say that people are sick, they're sick because they have symptoms of being sick, but it's not because we know that they have COVID. They might or they might not. We don't know.
That's a very important point, because yeah, the question was like, are you feeling well? Are you feeling sick? Maybe their allergies might be acting up and they mark like they're sick, and so getting the data would be really, really good. And also, since this isn't nucleic acid based, going back to what Ren said, these are tests that could be done on the spot.
Right, and then maybe one of the things that we're doing right now with the data that we do have is we're looking at the information that we get from the survey, and we correlate it to the actual number of cases in each of the regions, and we do see that we can have some prediction based on the symptoms. So some symptoms are more important than the others, which clinically it's well known, but we now have kind of a mathematical formulation for this, and basically saying just kind of a combination of the symptoms and information about other information about the patient could give you some idea of whether this person is more likely to have COVID or not. And it seems to work better than basically just looking whether this person has symptoms or not. Actually much better at this point. And this information is going to be, or these models are going to be much better if we have better data to train our models. Right now, we can only train our models when the kind of the ground truth is basically what we have from the patients that did get tested, and from the public data. So both of these are lacking either in terms of numbers or in terms of just kind of noisiness.
So from what you've talked about, is it too soon then, you're saying, to inform the actual medical researchers on, affecting what they're doing in the labs? Is there just not enough data, is what you're saying? Or is the data that we already have enough to do something or go a certain way on research?
I mean, enough data depends for what, right? So basically, if we want to, let's say right now we ask for about 7-8 different symptoms, for instance. And, we ask about the age and a few other questions. You take all this information together and you want to figure out whether someone is going to have COVID. You don't expect to be 100% accurate without an actual detection kit like Eran and Daniel are basically building. You're not going to get 100% accuracy on this, but you want to basically be able to have an idea in a population, let's say the population of a specific neighborhood, what is the percentage of people that are sick at that population, and you could potentially get to that accuracy, but not necessarily to the accuracy of the. Sorry, I have.
Okay. So while Eran's doing that, why don't you, Ren and Daniel, I mean, does this little, even though this little bit of data, I mean, do you just disregard it for now, take a look at it? You know, how would it affect what you're doing in the lab?
So this is regarding Eran's accumulation of data, right? That it doesn't really impact, maybe it might impact how we distribute, like, once we get this kit working, we'll go after like the hot spots and maybe try to target those first. I would say that might be one, but the other locations might not be hot spots just because people aren't filling out the form. So maybe it has to be taken with a grain of salt as well.
For my work, the small protein we are using to develop the detection reagent are very small, only 90 amino acid, and it can be produced in E. coli. So once we validate the method, if it works on that platform, we can scale up to produce enough reagent for millions of tests in a couple of days. So that's the attraction of this platform we're developing. And we are developing high affinity binding without using animals, without purifying B-cell clones from infected people. And we are developing the high affinity through evolution in vitro, and it can be done very fast. And this is the first time we're trying to develop this, so it takes a little bit more time and it requires some experience, which we are accumulating. And we hope it can be a platform for COVID-19, but also for a platform for other new emerging disease. And we can, in theory, if we do well, we can get it done in a couple of weeks.
Right. I think the scale-up point is really important because, from a paper-based diagnostic point of view, I mean, these technologies have been around, so the manufacturing know-how is there. So, if Ren's side can scale up, then everything should be able to be scaled up.
And then Eran, is it your, I would think that it's your intention to feed it, to obviously collate all this information and then feed it not just to UCLA researchers, but to researchers all around the country?
Yeah. So there's a few potential applications. So one application is basically to the hospitals themselves, and it doesn't have to be the UCLA hospital. It has to be essentially any hospital where we have enough information in their area. And so the idea is that if you want to understand what is going on, what is going to happen in terms of the resource allocation in the next 2-3 weeks, you have to understand how is going to be the influx of the patient in that hospital. So the number of people that are going to be admitted to the hospital. And that depends on, it doesn't depend on the patients that you currently have in the hospital, maybe slightly does, but basically you have much more information by checking what is going on with the entire population around that hospital. So this is, it's not specific to UCLA, but right now the best coverage that we have is in the LA area, which means that the hospitals in the LA area are going to benefit the most from this. But it could be, and actually one of the things that we want to add to this is another kind of part of the website where we're basically just going to ask the hospitals to add some information, and we're going to give them predictions that are specific to the hospital. So that's not implemented right now, but this is one of the things we're going to add next. And the idea is, again, if you're in a hospital in Boston and you want to know what's going to happen in Boston, so we can basically, give you these predictions under the projections that we have right now for the influx of patients. And if we don't have enough people enough people that filled out the survey in your area, then we could either get it from other predictions based on public data, or we can get it based on some collaborators that are, kind of more local in, let's say, in Boston in that case. And that would give some information for the forecast, because part of the forecasting is based on what's going to happen in the community, and the second part is what's happening in the hospital itself. So, for instance, how many people that are admitted are ending up in the ICU? How many of them are ending up in, with a ventilator? For how long, and things like this.
Mm-hmm. And I think Daniel had some questions coming up from the audience. So I think, Daniel, if you wanted to jump in and bring up some of those questions, you're welcome to do that.
Yeah, definitely. We're getting a lot, Eran, on just the survey itself, some specific details, and I'm going to combine a couple here. I think a couple of people want to know how many people have answered the survey, and what's that sweet spot number that you need to see a real impact. And then you also kind of just mentioned that you're using this data to forecast. So is the ultimate goal to use this to be predictive using a bigger data set?
Yeah, so let me start with the first one. So basically right now we have about 40,000 people that answer this. And it kind of stopped, a few days ago, the increase. And but today we're sending a follow-up email. So we're hoping that more people are basically going to give us information now. And actually now we also have more information that we give to people, because some of the feedback that we got from people is that they said, okay, what do I get from this? What's the incentive for me to fill this out? Which I kind of actually thought about it. I was surprised by that, because the whole reason I actually got into this is because I felt that there's a need, and we may not. I mean, the plan is not to publish anything. The plan is actually to kind of essentially stop COVID. But apparently this is not somehow universal, this feeling. So basically, some people think that it's kind of that I'm trying to get something by this. But so I should clarify: we're not trying to get anything for ourselves. We're trying to get something so that we will have a better control as a community over what's going on, without shutting down the whole, the entire country, which is basically what, we can always shut down the entire country and then we'll have a great control. But the question is if we can do things that are with a limited interference with our daily lives. So, one of the things that we're asking now in the follow-up, and basically people that come to the survey right now, we give them personalized information about their area, and we say essentially what, the grocery market, like I showed before, like the grocery market that you go right now, it's a place that has more or less sick people, and your area has more or less, and compared to neighboring areas and things like this. So hopefully this will also bring more people in. And then everyone that basically essentially is, all the participants, if you guys can fill it out and ask your neighbors to fill it out, we'll have more information about your neighborhoods, and we will be able to tell you something about your area. So yeah, so in terms of the applications, again, what we want to do, the first goal is to use this for, better forecasting. You can even just without doing any math, right? You basically can just look at what's going on, because we are following up on people, and also the survey responses themselves. It's not in one day. People gave it to us over, let's say, 2 weeks. So we have information, kind of a time series information. So you can see the dynamics of how things change. Maybe a week or two is not enough, but if you're going to look at a month or two, you'll be able to see how things change and they move from one neighborhood to the other. Which means that even without any kind of modeling, you'll have a feel of, okay, it's getting closer, it's getting further away from where you are. This is one thing. But the other thing is that we're going to formulate this mathematically and basically use the mathematical models that we develop in order to give you something which is more kind of rigorous, in the sense that will tell you what is going to happen in your neighborhood, and again, then inform the hospital. And then the hospital can use it in different ways. One of the ideas that we had, but this is obviously depends on the way, you know, each hospital wants to act on this. But, you know, if you have two patients that from a medical perspective, they have the same symptoms, they have the same risk factors, the same age, the same comorbidities, so, and things like this. But one of them lives in an apartment building that everybody is 80 years old, and the other one lives in an apartment building that everyone is 20 years old. You probably don't want to treat them the same way from a public health perspective. If you're worried about how the surge is going to affect your, you know, your hospital resources, then you have to think about it. If you basically send that person back home, and then the entire building basically gets COVID, it's going to affect you differently if everybody's 80 years old compared to everyone is 20 years old. So this is, of course, two extreme examples. But that's essentially one of the other things that we're looking at. But then we can also answer questions that are more, you know, specific to COVID. We're basically interested in looking at how, you know, temperature is affecting, you know, temperature of the area. And again, if it's only LA, it's less interesting, but we have people from all over the world, just the majority of the people that answered are from LA. You know, essentially the interaction between the different symptoms and the different demographics information and the exposure. And then when things are going to change, again, now things are kind of stable in the sense that most people are staying at home and people will go back to work, then we would like to see what is the effect of this? What is the effect of wearing a mask? What is, you know, in terms of the spread? And that could inform public health policies and basically other type of policies.
Right. So thank you for that. That was a very, very good, thorough answer to the question. I wanted to also bring back Daniel because we're getting a lot of questions right now about the saliva detection tests and things like that. So Daniel, you want to bring up some questions for Ren and Daniel?
Yeah. So I think the first was for Dan about his company, Phase Scientific. He's, from what I understand, the approach is extracting RNA. And I was hoping you could, at a high level for a broad audience, kind of dive into why this approach is either more sensitive or more accurate for extracting viral particles.
Sure. The conventional RNA extraction kit revolves around a solid phase technology, where our technology uses a two-phase system. So it's like an oil-water system, but both phases actually, they're immiscible, but they're predominantly made up of water. So they provide a mild environment for these biomolecules. So since it's a liquid-liquid extraction, you can actually sample a much greater volume, and therefore you're going to grab more of the virus, and that's where the edge is, part of the edge. Yep.
And so then going on the other side of it, we know that Dr. Ren's son is developing an antibody-based test. And Ren, I was hoping you could provide another high-level overview of why use an antibody-based test instead of using DNA or RNA nucleic acids, and why one is better or different than the other.
Yeah, I mentioned it very briefly, and I did not go into the detail on the difference between nucleic acid assay versus antigen assay. And the nucleic acid assay requires extraction, which Dan just mentioned. And then the next step is going to the PCR machine, which requires dedicated space and trained certified personnel. That will be a limitation. That is a limitation, and it will continue to be a limitation of how many sample a facility can test. And currently, the country is short of those capacities. And then there's another assay is detected antibodies. The antibody requires several days to produce. Usually it requires 7 or 10 days to produce antibodies, and before antibody is produced in the body, and you cannot detect it. So the antigen detection, the extraction is basically not much extraction process, and basically you can do it on spot, and so it will complementing the nucleic acid-based assays. Yeah, so the assay we're developing, we're using antibody mimic, which is simpler than antibody, and it's much smaller than antibody. It can be easily scaled up, and it can be produced at extremely low cost in E. coli. That's one difference between the our binder, we call it monobody versus antibody. Traditional antibody are much larger protein, and it requires immunization to generate, and it requires much more complicated facility, cell culture facility to produce, or animal to produce.
That's great. And I think a natural question or follow-up from that, and we're getting this as well, is kind of the status of these tests. And I think it's safe to say it's not ready to just deploy commercially, right now with the flip of a switch. So what kind of external resources are required from, from us, the greater biotech community, from companies, I guess from all three of you, that can help further your research along in any kind of like either licensing or funding phase, or any ideas that you have?
I can start. My lab studied the basic scientific questions: how virus replicates, how virus interact with host immune system. So it's the first time we try to develop a product for viral detection. And we moved extremely fast in terms of research in the laboratory. We got to work in the laboratory, but we are short of experience and a resource to make it robust, so can be scaled up, can be used in conditions outside of the laboratory. So that's the part we looking for help, and working with Dan is a significant step forward towards that direction. And he has experience and a resource on the lateral flow platform. And so thanks for Mark to link us together and to move it forward. But we're looking for additional conditions to stabilize the assay, to make the assay more robust.
So yeah, I agree with all the points Ren said. There's always, help is always appreciated. And I think at this stage, we are at the point where just started testing out, we started working together. As questions, come up, I'm sure input would be useful. Funding is always useful. I think in the lab setting, I think getting it to a point where it's working, I think we have a lot of expertise in manufacturing. I guess through the startup, I also have experience there too, but it's always good to get more information and try to get products out there to help people.
Yeah, I think from our projects, basically, what we need is data. So, to improve the models and to improve the predictions. And there's two types of data. So, for the Stop COVID Together project, basically, what we need is help in spreading the word that actually more data is going to be more useful in terms of all of the predictions that we're going to make. And again, more data is not only more data in terms of the number of people that participate, but also the number of times they participate, because we want to track what's going on across time. So this is one thing. So not, it's not a kind of a specific request, but I guess it is. So basically just spread the word and try to find ways of basically get more people to participate in this. So that's from this point. The other side, which is basically looking at the patient data, we're looking at the patient data in UCLA, the electronic health records in UCLA. And, luckily we didn't really have so far, huge number of patients, unlike other places in the world or even in the US, if you think about what happened in New York. So the numbers are relatively small, which is great in one sense, but from a data science perspective, it's limiting because then you have a smaller number of samples to look at. We have pretty nice results with this data because what we do is we work in a combination of data science and taking a lot of domain expertise from the clinicians. So it's the data science working together with the clinicians hand in hand and developing essentially the models together. But what would be really good is to add on top of this more data from other resources, from other hospitals, and just kind of combine them together. We have a couple of collaborations going on with other hospitals, but at least right now the data is relatively small. I hope it will stay small, but I think, in some places it is larger numbers. And, so one other type of help we could get is basically trying to get to this type of data.
So one of the questions that came up, this is a two-part question. One, in collaboration with the actual technology and using the cell phone. So what we had kind of kicked around the other day between all of you, between Ren and Eran and Dan, is, while you guys are doing the research on Eran's side, is there a way to talk about, putting an app together so the accuracy is better on the tests? And then the second part is, maybe using the GPS or location services to maybe help inform the data so it can scale up. So let's talk about the possible collaboration of what's being done in the lab and maybe working with technology to kind of create more accurate testing.
So, we've developed some scripts in MATLAB and other programming software to kind of take our images and turn them into numbers. So, there's definitely that translatability to a cell phone. We want to make these scripts so someone could just kind of look at it and by eye determine whether or not, they have COVID-19 or not. But, inspiring them to kind of use the cell phone app would help feed information to Eran's part of the project. So I think that's great. And I like the idea with the GPS, because then maybe questions about where they're located, you get immediately from where that cell phone signal is coming from.
Right. I mean, so again, from the perspective of getting the best predictions, that's the best thing we can dream of. Basically, that people will have every 5 minutes another test, and they will give it on their GPS, and we'll know where they are. But there's like some things that we need to consider when we do something like this, which is in terms of privacy. Right now, what we have is everything all the information that we get is under HIPAA. So it's HIPAA compliant. And once you get in, and actually most of it is anonymous, except essentially the email and your address. But the address, we actually don't ask for the specific address. We ask for the closest cross street or the zip code, something like that. When you go to the location, it's a little bit more sensitive. But I mean, of course, that would be really great to have, because then you could potentially model, for example, social distancing much easier than just asking a question. You can figure out where do people go, basically how much do they move around, and where do they, they can say, right now they can say that they live in one place in the survey, but actually be in a different place because they want to kind of do social distancing in a different town with their parents or with their family or something like this. While when you take the phone, you know exactly where they are, and so you kind of relate the COVID results, the person is positive, to the specific area where they're at. So it's definitely much more useful, much more accurate. And again, I want to emphasize that the issue with all of these modelings, and I'm sure you hear in the news all the time about different modeling of what's the number of people that are going to die in the US or in other places, and these models change every week, and maybe part of the blame is because of the models, but I think a lot of the blame is because of the data, because the data is not sufficient. So having cleaner data would help us for sure to get better estimations, and it's going to be very critical, especially if if you think about let's say the next wave that people are basically starting to talk about. Even if we go down now, then basically it might come back in the fall. Maybe it won't, which would be great, and then we can all go back to do what we did before. But basically, if it does, then we should be ready. And one of the ways to be ready is basically have better data.
And Ren, what how do you see, if possible, that the technology that exists could help what you're doing in the laboratory?
Yeah. Last discussion a couple of days ago between among three of us and Dan and Eran, we point out there is one possible way using cell phone to get the information of the test directly into the network by taking the image, which will provide more accurate information and also GPS localization. But today I want to bring up another possible collaboration is use cell phone directly as a detection device. Actually, many years ago, we collaborated with Professor Oskan and a postdoc in his lab, Qinsan Zhou, and working with a postdoc in my lab, and develop a cell phone based microfluidic device that uses a LED and a small microfluidic device, and it's attached onto the cell phone, can detect the virus directly and use antibodies. At that time, we use antibody, can see cytomegalovirus particles on the cell phone. So that's another possibility to detect the virus immediately, and then the cell phone actually provides the power, and the lens provide the detection directly. So that's another platform we can use cell phone.
Yeah, and maybe another angle to this is, so I guess maybe the numbers are not there yet, but in principle, when you look at a lot of these tests that exist now, there's always false negatives and false positives. And these could potentially be minimized by taking information about the patient that are outside of the test itself. So, if we look at the symptoms and other information that we gather from the survey, and we have some, kind of prior probability that that person might have COVID, that could inform essentially the results that you get from the test. So you could combine these two things together. So that's another way. It's not necessarily specifically to the phone. It could be also through the website. I think the phone itself can have two things that you don't have in the website. One of them is that it could be it's mobile, so you can basically know exactly the location, so the location information. And the other thing is you can potentially use the sensors in the phone, which I think is what Ren is referring to again either do something on top of the test and on top of the survey and inform the survey and the test, or just be part of it actually.
Great, thank you. I'm going to move it back to Daniel because we're about 10 minutes from ending. So Daniel, I wanted you to float up any other questions that we had from the audience.
Sure. Yeah, there's been a couple, and I know it's kind of an unfair question on in your guys' best estimation when you think you can have a diagnostic test on the market or kind of readily available. So, without holding you to it, if you could provide a little bit more information on that, we'd appreciate it.
So I guess from our end, we just started working with Ren on Saturday with the samples, and a student worked through the weekend, et cetera. So, but we're moving as fast as we can. There have been some challenges. I think that Ren had already mentioned about stabilization of the protein on different platforms. So, there's some fundamental studies that still need to be done. I think initially, I think both his lab as well as my lab, when we first got the samples, kind of wanted to just do a quick and dirty test to see if we could get some results. And I was trying to see if we get some results by today. So, I got some exciting news or not. And, definitely there are some fundamental studies that are still in the works, but, we are pushing as hard as we can. I think, Ren has shown with an ELISA at least that, this technology could work and his monobodies can work. So maybe Ren could talk about that as well.
Um, when we taking on this challenge at the end of January, and we was really rushing, and we try to get the product as early as possible. So now we, I mean, in our laboratory accomplished this, but now we need to go back to the beginning and to really sit down and redesign the protein. And one ideal thing, one unique advantage of our approach is we always can continuously improve the property of the binders, because we can modify them as we move around. So we think we need to go through another round of modification of the binder to make it more stable. We already have the affinity, but we need to make it more stable, more robust. So it probably, if it goes well, another 2 months, go through another cycle of optimization.
And I think one thing I just want to add is, talking to people in Ren's group and also talking to the students in my lab, I mean, they want to get something out there for the people. So I think that's been really nice to see. And everyone is gung-ho and trying to get their product out there to help, because, it kind of goes back to, if you're going to look for antibodies in people that have been infected, that comes late, and definitely depending on what type of disease we're looking at. Sometimes point of care assays will say you have to be infected for at least 3 to 4 weeks. That's too late. Whereas his target is you get infected and let's go after that virus right away. So we'll see. We'll keep you updated.
Thank you. And Daniel, do you have any other questions?
So those are pretty comprehensive. We're getting a lot of questions on the status of clinical trials and some of the ones that are being run at UCLA and other ones throughout the world. And I just kind of want to comment that we will have follow-on webinars on these that are more vaccine and treatment approaches. So stay tuned for those in the next few weeks.
And this is Mark, and again, thank you for participating. And again, TDG is here to help facilitate, and that was the purpose of this webinar. So the more we can help with facilitating your collaboration, because even when between the three needs, when you say about the first diagnostic point, the POC test, there's a lot of ways to approach it, such that, as trade-offs between sensitivity and specificity can make a big difference of saying, well, what's good enough to launch that may help Eran in one way, where it's like, okay, great, it's high on as far as false negatives, but still can make a difference with really helping to facilitate the tracking.
Yeah, that's a really good point.
And Mark, one question that came up is, not a lot of people know exactly what our division does, UCLA Technology Development Group. So in regards to that, one main question was, what is the role in regards to collaboration on this research, bringing people together? What other things happen in our division between researchers at UCLA and outside?
Well, and that's a really good question. I think one thing that's that might not be a low level of awareness is that many times collaborative breakthroughs are really through these type of introductions, because it's really finding people who are working side by side, which UCLA is known for, such a small footprint but highly collaborative. But at TDG, what we've done, especially with Amir Nyberg, who's our CEO, we try and help facilitate at different levels. So for instance, whether it's an industry-sponsored research, which is funding in the PI's lab, that's great. We're here to identify those companies, make the introductions, and try and align so there can be an agreement there. But similarly, if it's licensing, it's sort of a form follows function. We're here to facilitate whatever it takes to translate your research into commercialization. And so through these introductions, and then making sure the agreements that are put in place, again, to facilitate commercialization, that's really our role.
I should add, since the end of January, Mark, representing the TDG, has been working with my lab very, very closely, and I'm extremely grateful for the support and the guidance from the TDG.
I second that. You know, Mark and Earl connected me to Eran, right, and it sparked this whole area of research. In addition to what's going on in our lab, so you know, thank you as well.
Well, you're welcome. And thank you, of course, for your investment and time and effort into this critical endeavor to address this pandemic.
Thank you, guys. So I want to share on our ending screen here. I want to thank all the researchers for taking out the time from their very busy schedules to help us and help inform the greater LA area about what's happening in the research world. And we really appreciate your work. We hope that you are able to stop this pandemic from your good work. And I want to thank everyone at UCLA Technology Development Group for supporting this series. And that's it. So just so everybody knows, this series will continue with different types of themes. I'm just going to bring up my slide here as we end here. So we have a few more things coming up in the future. So the best way to find out about it is join our e-news or visit our website. And this will also be has been taped, and it will be up on our website and our YouTube channels. So thank you again very much to all the researchers and to all the people who attended, and stay safe out there. Thank you.
Thank you.